Uh, Again, Professor Wang, wonderful presentation and
Thank you.
beautiful, amazing videos and images. I'm definitely fascinated by them. I'm sure it intrigued a lot of our audience members as well. I would like to thank Dr. Sarmishta, for curating all the questions, and I'll try to take as much as possible in the limited time we have. So starting off, Professor, so with regard to the bubble propulsion mechanism of active colloids, is there any influence of the size of those bubbles on the movement of that micro or nanobot?
Okay. Well, before I answer this question, I want to thank everybody for listening to the talk, for all these interactions. I see a very active audience, amazing audience. And especially for those who are here for the first time, I encourage you to attend more of these ACS Science Talks. It's wonderfully organized. It's one of the best organizations I've ever seen for these research talks. Thank you so much. And to get back to Professor Mato's question, so do the size of the bubble affect these active colloids? In particular, I think that only affects the active colloids that are driven by bubbles, and that's one type of chemical propulsion. And there's actually a lot of chemical propulsion that doesn't generate any bubble. But for those that generate bubbles, yes, the size of the bubble and also the frequency of the bubble and where the bubble is generated, they all affect the particle's motion. And there's some theoretical analysis, some theoretical studies on that, but it's beyond my research, so I can't really answer very properly.
Thank you, Professor Wang. I'll move on to the next question. Can you elaborate on the mechanisms underlying the movement generated in Janus particles, like the one you showed, PMMA and AG microparticles? And
Okay.
does the coating have a role to play in the generated movement?
Okay, thank you. Okay, all right. I guess I'll just verbally explain. I don't have my slides here, but yeah. So first of all, the coating definitely matter, right? Otherwise we wouldn't need to put the coating on. So the coating is made of silver, and the material is very, very important. So silver, it oxidizes in hydrogen peroxide, and then the oxidized chemical, the silver chloride, gets reduced back to silver when you shine UV light. So this oxidation reduction is critical for the particle's motion. So the coating is really, really important. And then why does this oxidation reduction move the particle? That's another question, which I didn't really talk about. Simply put, if you have a chemical gradient, a local gradient, and this gradient can do a number of things. It can generate electric field, because if you have more positively charged, if you have more cations on one side and more anions on the other side, and then the solution is basically not electrical neutral. So you have more positive charge on one side, more negative charge on this other side, then you have electric field, right? And if you have a local electric field, and remember, these particles are colloidal particles suspended in water, so they probably carry some surface charge. More commonly, they carry some negative charge. So if you have a negatively charged colloidal particle in a locally generated electric field, this is electrophoresis, right? So, but it's locally generated electric field, so it's called a self-electrophoresis. That's one very classic mechanism of how chemical gradient can drive a colloidal particle. There are other mechanisms, but a little difficult to explain. So maybe for people who are interested in these mechanisms, how chemical gradient can drive particle, there are a lot of references you can look up online.
Thank you, Professor Wang. I'll move on to the next question. So have there been any advancement towards clinical applications of these active colloid-based micro or nanomotors? And a follow-up to that was, are there enough chemical reactants available naturally in the biomicro environment to fuel these micromotors for real applications or real-world application? And what are the potential challenges associated with this? Thanks.
That's a great question. Great question. Well, okay, to begin with, I mostly do fundamental research. So clinical research or applications is not my strong suit, but I have been very interested in this progress. Yes, there's a lot of research in this area using nanomotors or micro robot for biomedical applications, for environmental applications, also for assembling microfabrication applications. And in particular, I think most people are interested in the biomedical applications because of the movie Fantastic Voyage back in the 1960s. So imagine you have a small submarine that's cruising inside human body, can treat diseases or do something funny to your body. And that's the dream. That's one of the holy grail of nanotechnology. And there's a lot of progress. Actually, I think a few years back, my lab published a review article on all sorts of challenges of using nanomotors or nanomachines for drug delivery in particular. Because if you think about these chemically active colloids or chemical nanomotors, I think the question is very appropriate. You need to find a fuel that's abundant in human body and also safe to human body. And there's not a whole lot of chemical reactions we can choose from. In my talk, we used hydrogen peroxide, we used UV light. So neither of this is really biocompatible or biosafe. But in our bodies, we have a lot of chemicals. We have water, we have acid, we have glucose, we have ATP. So people have been trying to use these chemicals as fuel to drive chemical nanomotors. There's some progress, but I think we still need to work harder on that.
Thank you, Professor Wang, for that answer. I'll move on to the next question. It's from one of the audience members. So self-propelled colloids exhibit randomness in structure formation. Is it easy to control that?
I think by structure formation, well, individually, these particles are moving randomly because they're affected by Brownian motion, right? So they're bombarded by water molecules all the time. And there are some ways to control them. The easiest way is to make the particle magnetic. So you can put some magnetic coating on the particle or start with some magnetic material. And then once it's magnetic, you can use a magnet to control where the particle is moving, right? So basically you have a joystick and you can control where it's moving, whether it's moving left or right. And there's a lot of research on this already. Other than using magnetic field, you can also use other external fields, electric field, ultrasound, light. I think my lab recently published a paper on using ultrasound to steer a colloidal active colloid. But I think the most interesting, most promising way is to use a gradient. So this is something called chemotaxis. In biology, our cells or bacteria, they control their motion not because they're magnetic, but because they know which way to go. And they do this because they move up or down a chemical gradient. So this is called chemotaxis. And if we can make an active colloid that responds to chemical gradient, that it can know which way is up or which way is down, then this is far more ideal than using a magnet inside human bodies. I think people are already working on this, but again, we need to work harder on it.
Thank you, Professor Wang. Just to build upon that answer, there was another question which said, like, how would you compare the various modes of driving the motion of these micromotors, light, magnet, acoustics, etc.? Does chemically driven micromotors provide any specific advantages of the other modalities?
Yeah, okay. I think chemical motors are pretty good if you don't want to interfere with them, right? So if you want just to leave them alone and they move on their own, I think chemical motors are very good for that because they can take chemicals from the environment and they convert it into motion and they just swim. Whereas if you use magnetic active colloids or electric active colloids, you need to supply the magnetic field or electric field. That can be a little challenging or undesirable. But chemically active colloids or chemical nanomotors are, in my opinion, not exactly suitable for biomedical applications because, well, I wrote about this in our review article because there's a lot of challenges. You need to find a good fuel and also some of the mechanism to drive them are not biocompatible. Whereas the magnetic motors or electric motors or ultrasound motors are more biocompatible because you can find ways to supply this energy without hurting human bodies. So in this sense, I personally, I'm more in favor of using, let's say, ultrasound motors or magnetic motors in biomedical applications. But I think things are changing. So I think there's a lot of progress in chemical motors. Maybe someday there will be a huge breakthrough and people will develop good chemical motor that can do all the job.
Thank you, Professor Wang. Coming to the next question, so this is related to water treatment. So say, can these micro or nanomotors be designed in such a way that they can consume the pollutants in water, or they move toward, say, that pollutant, use that as the fuel and move towards more pollutant? Say, for example, oil spills. So it can just consume oil and keep on moving and consuming oil. That way we can clean oil spills.
Okay. That's a great question and actually great idea. But there's actually a lot of studies on using these nanorobots to clean up water. So for example, the research lab of Martin Primera, they are one of the pioneers in this research area. They have published a lot of papers in this topic. They design all sorts of chemical motors made of all sorts of materials. And some of them can degrade pollutants because they are, let's say, photosemiconductors. So when you shine light, they generate holes, and the holes are really oxidative, so they can degrade organic pollutants. And some of them are just attract pollutants. So if you have microplastics, and these motors may go next to a microplastic and bind to the surface and move away. So basically, they absorb the microplastic and clean it from the water. But I don't think I've seen any research on using the pollutant as a fuel and then jump to next pollutant. I think this is interesting thought. Maybe there are already research paper on this, but I don't know. But one challenge is to really make tons of nanorobots very, very cheaply so that we can just spray them into the ocean, right? Because the ocean is so big that we need to make a lot of these nanorobots very, very cheaply to really be cost effective.
Thank you, Professor Wang, for that answer. And I hope like someone is working on that idea that you're talking about. So, Krishna, we are almost on time. Is it all right if we take a couple of more questions?
Yes, Ashish, feel free to take a couple of more questions.
Sure. Thank you so much, Krishna. So coming to another one, is it possible to prepare active colloids out of drug particles or drug molecules, and then that can be used for targeted therapy?
I think so. I think people have already done that. But again, I don't do that in my lab, but I think it's a valid idea. Yeah.
Thank you, Professor Wang. Another one from the audience member, is it possible to study the plant growth patterns and response to certain enzymes using nanobots? Again, I know this probably is not your field of expertise, but question nonetheless from the audience.
What is the question again? Sorry.
I can repeat that. So is it possible to study the plant growth patterns and response to certain enzymes using these nanobots?
What is plant growth patterns? Like how trees grow?
I think so.
Okay.
Someone wants to monitor that.
Okay. Well, okay. But I think in general, it's a little bit challenging to use nanorobot or nanomotors to study something really, really big. Because, you know, in terms of scale, they are so far apart. And also it's because we usually need to use a microscope to see these nanomotors. But if you are examining something macroscopic, then it's difficult to do that in the lab. But there are studies that mix these synthetic active colloids with actual living active colloids like bacteria or cells and see how they do to each other, like how the active colloids will affect bacterial growth and vice versa. This goes to the topic of hybrid nanorobot. So there are actually people who combine bacteria with a nanorobot, sort of like a like cyborg kind of thing, right? And that will have the beauty of both species. It will be like a living creature, so it can take energy from the environment, but it also let's say it's inorganic, so it's more tolerable to harsh conditions maybe, or it has a drug molecule in it. So I think the idea of hybrid nanomotor or hybrid nanomachine is pretty good.
Thank you, Professor. I'll just take one more question and then you can give your final thoughts after that. So I'll just take this one. So one of the audience members asked, what was the methodology that you used to capture those videos of active colloid motion? If you can provide some insights on that.
Okay, I can, but I think it's better to refer to our recent paper published on ACS Nano. We had a very, you can call it like an educational paper or tutorial paper that talks about how you record these videos, how you analyze the videos, how you draw these trajectories, or how you draw these pictures, and the challenges and the mistakes you might make. I think it's published 2 or 3 years ago on ACS Nano. So for people who are interested in these technical details, please refer to that paper.
Thank you, Professor Wang. That'll be all for all the questions. Thank you.
Great. So am I supposed to say something?
Yes, Professor, it would be good if you could provide some thoughts for the wider scientific community, because you know that a lot of us are graduate students and early career researchers. So your thoughts around how the field is going to progress, and some of your insights around, what are some of the potential research areas that can be opted by them, to build upon, more scientific content in this field, and then again get some publications out.
Okay, great. Okay. So I joined this field about 15 years ago when I started my PhD at Penn State University in 2008. So that was at the beginning of all this research on nanomotors. And 15 years later, I think it's growing into a very nice, very active research area. It's not a huge research area. It's small compared to, let's say, solar cell, but it's very active. And it's very interdisciplinary because people from soft matter physics, chemistry, engineering, material science, all joining forces to understand what's going on, because you can probably see there's a lot of mysteries, right? There's a lot of interesting things that are going on. But also people care about how to control them, how to make them useful. And I think there's 20, even, we're organizing this symposium to dedicate this, to commemorate the 20th year anniversary of the first discovery. Even after 20 years, there's still a lot of unsolved issues, a lot of open questions. So the paper, the perspective article I recently published on JAX is precisely about that. So it's about open questions on chemically active colloids. And there's a lot of open questions. So that means there's a lot of research opportunities for grad students and also for faculties like me. And so I think to me, this is a very good time to jump into this research field because we have some basic understanding. We have a very vibrant research community. We are made of really good people, so we're very friendly, we're collaborating with each other. And also a lot of open questions, fundamental questions, applied questions that are ready for people to just explore. So to all these young students or postdocs out there, I really welcome you to take a look at this research field, read some papers, and see if you're interested. And for senior faculty members who are just looking for ideas, I think the nanomotors are really good model system to understand a lot of complex systems. They're also good, call them tools. So we can use nanomachine to, let's say, do biomedicine or solve environmental problems or do fabrication. So this is something, even if you're not doing nanomotor research, you should take a look and see if you can apply to your own research area. That's all for me.
Thank you, Professor Wang. That was wonderful. And I'm sure that our audience had pretty much a lot to take away from this. So this brings us to the end of today's session. We hope that you enjoyed the broadcast. I personally did a lot. And we invite you to view the edited recordings from the past events in the ACS Science Talks library that's available on YouTube. On behalf of all of us at ACS, thank you so much for joining us. We hope to see you soon for more ACS events. Until then, stay safe, stay healthy.
Right.
Bye-bye.
Thank you very much.